this again. Sorry, share screen. Now we can do it. It's all right. Take your time. We haven't started yet. There we go. Cool. Okay. So this is the second revision lecture. Don't worry about the numbers. It was the old numbering system. So in this lecture, we're going to look at 5 different things that we looked at in the later part of the lecture series. So we're going to look at how Cre recombinase actually works, and because many people have asked about those Cre-LOX-P experiments and the reporter, the Rosa mouse. So we're going to go through that in a little bit of detail. Also had questions about genetic complementation. Again, people always seem to have a little bit of a problem with the genetic aspect of those Nusslein-Volhard screens. We're going to look at ES cells and teratomas, just make sure we're all up to speed on that. Then we're going to go and look at stem cells and transit amplifying cells and look in that in more detail and look at things that people have found a bit complicated about that. And then we're going to look at Chick and mammalian gastrulation, because there's been lots of confusion from me and others about the epiblast and where gastrulation occurs and these sort of things. So, to start off, we have a question. Cre is a DNA recombinase. So have a think about that. And as you can see, most people last year answered that as true. Cre is a DNA recombinase, and that's central to its ability to function and how it makes these inducible knockouts. And here's a related question. CRISPR-Cas protects against what kind of viruses? So here we have a selection of viruses. Have a think for a minute. Sorry, you can't hear me. I had all the wrong setup. Okay, I'll whip back quickly and talk about everything we're doing. But that was bacteriophages. Okay, I'm really sorry about that. I had the wrong plug-in. So this is our lecture plan. Cre recombinase, we're just about to start looking at genetic complementation in the Nusslein-Volhard screen, ES cells and teratomas and other aspects of their biology, stem cell and transit amplifying cells, and Chick and mammalian gastrulation. So we know Cre is a DNA recombinase, we know that, and we know that CRISPR-Cas protects against bacteriophages. Now, why is that important? Well, Cre recombinase is actually an enzyme that the bacteriophage uses to integrate into the bacterial genome, and it modifies the virus genome, allowing it to integrate by these recombination at LOX-P sites. So so it's a viral recombinase. It cuts up DNA, the virus genome, and integrates it into the bacterial genome. And that's the way that bacteriophages can survive in, or at least their genetic makeup can survive in without drawing attention to, for instance, the CRISPR system. So what is a LOX-P? So LOX-P is the LOX site for what's called the P1 bacteriophage. So this is a kind of bacteriophage, and this is the specific site that this bacteriophage uses, and Cre can cut at these and recombine at these places. So what can you do with these LOX-P sites? So here are a few things.
So wherever you can cut DNA in a certain place, you can move the DNA about. So the first thing we can do here is excise or insert DNA. So we can remove DNA just by cutting with LOX-P sites. Typically, you cut the part of the DNA that's between those two LOX-P sites, and that's typically what happens in a Cre-Lox knockout. Other things can happen. You can invert the DNA that's within those two LOX-P sites. We can translocate DNA from other locations. So if you have 4 LOX-P sites and you can cut out pieces of DNA and then you can flip them round. And so that's translocation and cassette exchange. But typically what happens, what we're interested in our transgenic and knockout models are the excision and insertion, and also this cassette exchange. So the excision and insertion is very important in forming knockout animals, and this cassette exchange is really important in forming what we call knock-in animals. So here we have the excision insertion leading to disruption of a gene and knockout. So Cre-Lox removes the DNA from between those LOX-P sites. So we have our gene B and all of its exons here in black, and the construct has LOX-P sites flanking the second exon and those 5 other exons. And so what happens is the Cre recombinase cuts at these LOX-P sites, removing that piece of DNA from the B gene and then making it a truncated form, which is probably non-functional. So this is what typically happens when Cre-Lox is activated in a cell. So normally that LOX-P flanked region is what we call a floxed region, flanked by LOX-P, is in all the cells in the mouse. Now, typically, the expression of Cre, which is what cuts DNA at that LOX-P site, is tissue specific. So what we have here is on the right we have a mouse carrying a floxed allele. So here's our allele, here are our LOX-P sites, and it's expressed in every cell of the mouse. In the other mouse we have a Cre recombinase under the control of a heart-specific promoter. You mate those two mice and what you get is a mouse that has floxed alleles throughout and also the ability to express the Cre recombinase under that heart-specific promoter. So in the rest of the mouse there's no Cre expression, there's no removal of that DNA from the floxed site. In the heart, however, where there is Cre expression, we get removal of that blue gene from that region, the heart only, because there we have the Cre and we have the LOX-P sites. So we can make these Cre not be on all the time in the cells it's expressed in, but rather we can induce the Cre, and this allows for time and location-specific recombination. So our location-specific recombination is controlled by what promoter Cre is controlled by. So a heart-specific promoter would express Cre in the heart. What we can do with this Cre-ER, which is a nuclear receptor called the estrogen receptor, and that's bound, that's a fusion protein to the Cre recombinase, and what this does is it allows control of the subcellular localization of Cre. So without tamoxifen, Cre-ER is in the cytoplasm. Once it binds this drug tamoxifen, it can travel into the nucleus and then it can cut at these LOX-P sites. So here we only have LOX-P cutting wherever Cre-ER is in the nucleus. So the nuclear receptor is cytoplasmic unless bound with tamoxifen, and then the Cre-ER fusion protein is able to travel into the nucleus and cause that removal of that gene of interest. So the more complicated uses of Cre-Lox technology, and we talked about these in those fate mapping experiments, is how we can use LOXP to activate a mutation and mark it at the same time. So what we have here is a LOXP flanked stop domain in a transgenic KRAS with a mutation that makes it oncogenic. But while we have that stop in there, the KRAS can't be fully transcribed. It's a stop codon. It will prevent the transcription of that KRAS. In this case, when the LOXP sites are cut out by Cre, this is removing that stop, and it actually restores the normal genetic structure of that KRAS, and then we get KRAS G12D activated KRAS only where Cre is activated. And we also have a reporter gene here which tells us exactly where that mutant KRAS is activated. And what we saw there in this experiment is wherever that mutant KRAS was, we saw a tumor. So without that control, without that stop LOXP in there, you would just get KRAS expressed everywhere, and the mouse would likely die in utero because of how important RAS is. So when the Cre-ER is induced by tamoxifen, this removes that stop codon, and this allows the translation of the mutant RAS. So this is a site-specific inducible mouse tumor model using this LOX stop LOX technology. And so we come to even more complicated uses of Cre recombinase, which relies on the random nature of how Cre recombinase works. And so this is the Brainbow mouse, which we talked about again in that fate mapping lecture, where we have an array of different fluorescent proteins all flanked by LOXP sites. Then when Cre is active, a number of those LOXP sites are cut, and there can be recombination or loss of those fluorescence genes. And so we will always get a random combination of these four fluorescent proteins. And what this means in practice is you get a number of different colors where genes are, where cells are expressing 1, 2, 3, or 4 of these fluorescent proteins, and that allows a wide range of colors that we see in the Brainbow mouse and also the Skinbow fish and a number of other of these kind of multicolored tissue labeling fate mapping processes all use this random cutting of LOXP and random expression of fluorescent proteins. So here's a question. In a keratin 14 promoter Cre-ER LOXP stop LOXP GFP mouse, what happens when you add tamoxifen? This is a complicated one, so I'm going to leave you to think about this, but think about the promoter, think about what Cre-ER is doing here, and think about what it's expressing. And we'll go through this in a little bit more detail. So what happens when you add tamoxifen? The epidermis expresses GFP. So last year your cohort got this right. So basically what we've got here is keratin 14 is only the promoter. So keratin 14 is controlling where GFP is being expressed. So it's GFP that's being expressed. So in the presence of that stop codon, GFP won't be translated. When we add the tamoxifen, the Cre-ER will cut at that LOXP, removing that stop, allowing GFP to be expressed. So that's what happens when we add tamoxifen. So when you see these kinds of questions in the multiple choice or in parts of exams, always look at what the promoter is. so keratin 14 promoter, what's in between those LOXP sites, so LOXP stop, for instance, and then what's being expressed under the control of that promoter, which in this case is GFP, or green fluorescent protein. So let's look at this idea of genetic complementation, particularly in the context of that Nusslein-Volhard screen. Okay, so remember what happened in this Nusslein-Volhard screen, where we had wide-scale chemical mutagenesis which induced random changes and deletions throughout the genome. And then Nusslein and Volhard looked for embryonic lethal effects, so effects that caused defects that prevented the adult form of the organism. And then we back-crossed into what are called complementation groups. So we're going to look at what back-crossing into complementation groups really means. So the idea is, if you have two mutant flies that have the same phenotype, it's possible that the mutation could be in the same gene, or it's entirely possible that the mutation could be in different genes, even though they have the same overall defect. So are the mutants in the same allele? Are the mutants homozygous? And so what we can have here is, if you cross two mutants and they have homozygous mutations in the same gene, so in this situation over here, so both mutant 1 and mutant 2 have the mutation in the same locus, what would happen is, if you crossed these two together, we would still maintain a mutant phenotype. However, if mutant 3 had a homozygous mutation in locus A and mutant 4 had a homozygous mutation in locus B, when we mate those two together, because then they would be heterozygous for each mutation, what would happen then is you would see normal phenotype. So this way you can compare whether genes are in the same complementation group, i.e. you cannot cross them to rescue the phenotype, or whether they're in different complementation groups where, even though they have the same phenotype, they look the same. The genetic mutation, the gene that the mutation is in, is different, and therefore they are in different complementation groups because you can rescue the phenotype by crossing. So this was the way that mutants were divided into complementation groups. So when you see this idea of complementation groups, what they're actually saying is the same locus, and therefore most likely, except in occasions where these mutations are very highly linked, very close to each other in the genome, this typically means that the mutations are in the same gene. So we have the same phenotype, same gene mutated, then all offspring are mutant, right? Complementation can only occur when the mutations are on different genes. So in that previous case, both the mutations were in gene 1, and therefore all offspring from these two flies will be mutant. There's no complementation. Complementation occurs when mutations are on different genes, and here we have the first fly which has a mutation in gene 1, our second fly which has a mutation in gene 2. Therefore, in our first filial generation, our first generation from this cross, all the flies will have one mutation in gene 1, one mutation in gene 2, and therefore they'll all be wild type, as these two mutations complement each other. Let's move on to ES cells and teratomas. So here's a question. Embryonic stem cells are found in which part of the embryo? And I'll give you a little time to work this out. Of course, almost everybody got this right. I'll go through. So it's the inner cell mass. The embryonic stem cells are from those pluripotent inner cell mass. Remember, the troph ectoderm makes the embryonic, extra embryonic tissues such as the placenta. In the morula, all the cells are totipotent, and primitive endoderm is something I made up. So remember, there's always that thing I made up and the things that can't possibly be the answer to the question. And that's how you work out my multiple choice questions. So embryonic stem cells come from that inner cell mass of the blastocyst in mammals. So we have our blastocyst, we have our inner cell mass. This is our troph ectoderm, which becomes the placenta. Our inner cell mass, we can take those cells, grow them in culture and form these embryonic stem cell colonies, which we can't do with troph ectoderm. And it's those embryonic stem cells that can form all cell layers. And if we implanted those colonies into a nude mouse, we would get multiple teratomas. So teratomas are tumours of multipotent cells that can spontaneously differentiate into all the cell types. So here's our teratoma. We get the endoderm, which is adipose tissue. So we've got a lump of fat here in our tumour. We've got some mesodermal tissue. We have muscle. There's also bone, which is also mesodermal. And then ectoderm, we get skin, hair. Occasionally you get teeth in teratomas. And this is proof of pluripotency. So these teratomas are ES cell tumours. And if we implant ES cells into nude mice, we get a similar thing occurring. And here it is. We inject those ES cells from mice. They form these teratomas. We can see this in the nude mouse. Here's our control. And here was that GPC4 mutant mouse, which is unable to differentiate into the different different cell types. We can see here much larger teratomas, and the teratomas contain a range of different tissues. The tumours from the GPC deficient mouse have far less specification of tissues, so they're unable to differentiate into these different cell types. And remember, embryonic bodies are teratomas on a plate. We get those embryonic stem cell colonies. We grow them for 6 or more days in culture conditions. These embryonic bodies form, and if you take a section through these embryonic bodies, you will see ectoderm, mesoderm and endoderm, but in a kind of unarranged way. So can you control embryonic body differentiation with signaling proteins? Let's think about this. Here's a question. This secreted protein specifies epidermis from the ectoderm. Give you a bit of time to think about this. So yeah, when this question was answered, there was a difference of opinion. Definitely wasn't EDA. That's ectodysplasin, and that's to do with the formation of hair follicles and adnexa. Remember, Sonic Hedgehog and Wintate are not specifically required to make epidermis from ectoderm. Sonic Hedgehog is required for neural tissue. Remember holoprosencephaly, and the dosage of Sonic Hedgehog is very important in that. Remember that Wintate is required to specify some, or Wints are required to specify some of the tissues, the neural crest, but typically are inhibited to make the head. But we require BMP4 specifically to make epidermis from ectoderm. So BMP4 is both necessary and sufficient to make epidermis from ectoderm. And how do we know this? Well, we know that that ES cells can control their fate. Once they become ectodermal cells, they can either become epidermis or neuron. And dependent on the signaling they are exposed to, or what the ES cells are exposed to, will go down an epidermis path or a neuronal path. And remember our clues from Xenopus development, where BMP4 inhibition by caudin, noggin and follistatin is very important to induce a neural fate, in the ectoderm, but our BMP4 is required to make ventral ectoderm into epidermis. So if we activate, if we express P63 in ES cells without BMP4, this induces epidermal keratin expression. And if we treat ES cells with BMP4, we can get cells in our colonies that express keratin 14. We can see that here when we just express P63, we get an increase in keratin 14 as well. So not only is BMP4 required, but P63 is downstream of BMP4 and is necessary and sufficient to drive ES cells to an epidermal fate, even in the absence of BMP4, which is what we see here. So BMP4 induces P63, and then it's P63 that induces the epidermal fate, so keratin 14 expression in our keratinocytes. So P63 is downstream, but can also be necessary and sufficient to drive ES cells to an epidermal fate. So let's look at stem cells and transient amplifying cells. There's this initial concepts that one cell could give rise to many progeny in the epidermis. And in the mouse epidermis, we have this columnar arrangement of cells called the epidermal proliferative unit. This is a very old concept. It's older than me, which makes it nearly 50 years old. That this central stem cell is surrounded by its progeny, which can themselves divide a limited number of times, and then once they move from the basal layer, once they delaminate from that basal layer, then they can differentiate. They are no longer competent to divide, and they can perform terminal differentiation processes to make the rest of the epidermis. Again, one cell can give rise to many progeny, and in the intestine, the villi is huge compared to the crypt, which contains the stem cells. So there's no one-to-one relationship here. The cells in the crypt must be producing an awful lot of cells to be able to contribute to a wild-type villus. So if we have stem cells giving rise to differentiated cells directly, we'll have too few cells to be able to homeostatically maintain a tissue. So at best we'll have a one-to-one ratio of stem cells to differentiated progeny. And remember, in that more modern interpretation where we just track cell fate, this one-to-one ratio is that balanced system of cell division where one dividing cell gives rise to one progeny and one terminally differentiating cell. So we need that intermediate population where a stem cell gives rise to 4 differentiated cells through an extra cell division. So typically these transient amplifying pool of cells, or transient amplifying cells, divide a limited number of times to provide amplification of that cell population. So the cell population is transient because it can only survive for a few cell divisions, and it's amplifying because it increases the number of cells in the final tissue. Transient amplifying cells, or transit amplifying cells. So these are the product of stem cell division that can themselves undergo a limited number of divisions that finally give rise to differentiated cells. Let's finally look at gastrulation in different organisms, particularly higher organisms as like mammals and chick. So remember that unlike Xenopus or Drosophila, once we get to higher organisms, then the initial structures tend to be flat. So in chick development, the embryo sits on the yolk a collection of cells called the blaster disc. And this blaster disc forms, and from this blaster disc, all other tissues are formed. Remember that concept of the bullseye, which tells you that an egg has been fertilized. This concentric circle on the yolk shows you where the blastoderm is, and that blastoderm is what will become the chick. So remember, this is flat, same as with zebrafish. It's flat on a circular, on a round yolk. So very similar to zebrafish. So zebrafish is flat on a round yolk. The chick has a flat embryo on the round yolk. And remember that in chick embryos, that initial axis specification, i.e. what is anterior and what is posterior, is by gravity. So the egg gland or yolk rotates once the egg is laid. Lighter parts of the yolk push up the blaster disc, and the uppermost part becomes the posterior, the back end of the embryo. So we can see this here. The yolk rotates in the shell. We can see here that the top becomes the uppermost part becomes the posterior end, and the part closest to the ground becomes the anterior end. And this governs where our primitive streak, which is the area that gastrulation occurs in, will be occurring. So that chick organizer is the primitive streak. And the primitive streak is in the posterior marginal zone of the blaster disc. Again, this is where all cell movements are started. And so if you transplant the posterior marginal zone to another region of a developing chick embryo, we'll get an additional induced primitive streak. Now, bear in mind that typically, like for Xenopus, when this experiment occurs, we take the dorsal marginal zone. So we're taking this is posterior as opposed to anterior, rather than dorsal as opposed to ventral. But that's where the involution, all these cell movements that comprise gastrulation, begin, and so that's where the organizer is, and that's what you need to transplant to make a second axis. So this is organized. So the organizer in chick is called a Henson's node, and the Henson's node moves anterior to posterior, even though the initial involution is posterior. So the primitive streak is formed posteriorly, but then Henson's node moves anteriorly across the developing chick. And so, as we've shown before, we get this concomitant thing where, or things happening at the same time, where we have specification of tissues by the node, while at the same time, more anterior, we already have neurulation. So what happens is Henson's node moves down the embryo, and so we already have head tissues specified with full neural tube closure and full somatogenesis in regions that are complete in their differentiation, compared to the closer to the node or more recently involuted, and we have here neural cells just being formed and the tissue layers just being specified. So it happens, different things are happening in different parts of the tissue. This is different to, say, Xenopus, where we get gastrulation completed and then neurulation happens. So in higher organisms, such as chick and mammal, these things happen simultaneously in different parts of the embryo. So if you transplant that node, so now we've gone beyond the primitive streak, we've formed that node that's going to be moving anterior posterior. If you move that node to a different region of the blaster disc, you get a second complete induced axis in the host. So you can see this here. We've taken our Henson's node and we're grafting it into a donor, which itself has a Henson's node, and it's moving down, and we get a second induced axis where that Henson's node, or where that organizer, has been transplanted to. The mammalian embryo, as we know, develops from the inner cell mass. The inner cell mass forms a region called the epiblast, and it's the epiblast that's important in the formation of the embryo, so in gastrulation and further on in neurulation. So the primitive groove develops in the epiblast, and again we see this delamination of the epiblast, so cells moving from the epidermis and then moving into the inside of the tissue, and then it forms mesoderm and endoderm, which is exactly the same as the chick. So these are the key things you have to sort of remember in terms of progress of these things, is that in Drosophila we start ventrally gastrulating, and whereas in Xenopus we see gastrulation from the dorsal part of the embryo, and that's in a round embryo, so we get different formation of the layers in, say, zebrafish, which is the first flat embryo we looked at, and that requires that involution and moving back underneath the tissue. And then when we get to chick and mouse, we get this delamination that's required as part of the process of gastrulation. So just be aware of the different major forms of cell movement during gastrulation processes. So that's it for this lecture. We looked at Cre recombinase, and we looked at in particular how using Cre recombinase and those LOXP sites enable you to do different, quite complex things, including tissue specific expression and also inducible expression using Cre-ER. And then we also that stop codon, which you can insert in the LOXP to prevent the expression of a transgene. And we saw that with our RAS and the example we had for the question of GFP with those the floxed stop codon. Then we looked at genetic complementation, and in reality a complementation group is just the same gene, so that's the easiest thing to remember there. We looked at ES cells and teratomas, and we looked at stem cells and transit amplifying cells. Think of those as further and further along that stem cell potential pathway. We looked at how ES cells give rise to teratomas, which have all of the different tissue types, ectoderm, mesoderm and endoderm, and also how we can drive ES cells to a particular fate by using signaling pathways and activating signaling pathways we know induce certain tissues in our model organisms. So BMP will induce epidermis in ES cells. We looked at stem cells and transit amplifying cells briefly, and the concept that you need a transiently amplifying population to be able to replenish a tissue fully. You don't just need stem cells. Although I will say that in epidermal keratinocytes it seems that this mode of balanced versus proliferating cells seems to be all that's required in the epidermis. And then we looked at the different ways that chicks and mammals gastrulate compared to, say, frog and Xenopus. So I'm going to leave it there, and like I said, well, I said but you didn't hear, so I'm going to rephrase and say again. I would typically have a session where anyone could ask me anything in the lecture hall. Obviously I can't do that, but I noticed that not very many people attended the office hour earlier on today. So what I'm going to do is have a final office hour that goes on until 2 p.m. So that would be the end of your lecture. Typically, I give you permission to stay in the lecture hall and use your electronic equipment, or you can go home and use your electronic equipment there. I do also appreciate that you've got your project write-ups due, I think, at the end of this week. So yeah, just use that time how you want to use it. but I'll be available to answer questions until 2 p.m. Okay, and with that, I'm going to say thanks. It's been really great. It's been really great to lecture to you in this weird, remote, 6,000 miles away. I'm really surprised that we actually managed to do it every single time. I was really impressed with that. And thank you for being here, and thank you for being so attentive and asking questions. Still, if you're confused about anything, do ask me questions by email. I'll be available by email beyond the office hours. And I just want to say have a good rest of the year learning things. Also, best of luck with not only my exam but also everybody else's exam. I just want to say thank you, and it's been great fun, and good luck with your exams. So I will sign off there. My office hours starting. Ask me anything related to the lecture course, and yeah, hopefully I'll hear from some of you in due course. So I'll see you. Okay, bye. Thank you. That's really embarrassing, but thank you so much. Yeah. So, like I said, you can just ask me questions for the next hour and a half, shall we say, and then always you can contact me by email. But I'm going to sign off so you can do other things. All right. See you all. Bye.